Next part, it is related to female reproductive structure in the flower, that is gynoecium. Now, here we study in detail about the pistil, nothing but the gynoecium, and Megasporangium, nothing but the ovule, and embryo sac. So this concept we'll study in detail under the second part in pre-fertilization events. In the first part we studied the microsporangium and pollen grains. Here we study megasporangium in detail. Let us try to recollect some of the aspects, some of the concept already we studied in first PUC, morphology of flowering plants. So what and all we studied actually related to gynoecium part. You know that there is a difference between pistil and carpel. Even though the term is representing female reproductive structure, pistil term we use at one particular time and carpel term we use at another particular time. So what is the main difference? In general, to represent the female reproductive structure, we use the term pistil. But whenever it comes to the number of female reproductive structure present in the flower, at that time we use the term carpel. So that is how we will describe the female reproductive structure as monocarpellary, bicarpellary, tricarpellary and multicarpellary condition like this. Okay, now the question comes, the number of carpels in a flower, whether they are fused or free. If they are fused, what type of technical terms are used? Here you can see, suppose a flower consisting of only one carpel, then we use the term monocarpellary, single carpel. Suppose more than one carpel is present in a flower, then the condition is called multicarpellary condition. Have you seen any type of flowers where it produces more than one carpel? You might have observed this, but you have not keenly observed those parts. If you have observed the Michaelia champaka, there it produces many carpels, okay, and those carpels are not fused, they are all free, and that condition we call it as multicarpellary apocarpus condition. They are all free. So again there are two technical terms comes. When more carpels are present and they are all fused, then it is called syncarpus condition. Very commonly observed in citrus, where many carpels are present and they are all fused to form a single ovary. So we call it as syncarpus condition. When many carpels are present and they are all separate, then we use the term apocarpus condition. So these two are the conditions which commonly observed in some of the flowers like Michaelia champaka and citrus. In some plants it is only one ovary, then we use the term monocarpellary. Now each pistil has three parts. Now if I draw a rough diagram here, now this is the stigmatic surface. Here you can see the long style and the ovary. Okay, what is this stigma actually? What is their function? Stigma acts as a landing platform for the pollen grains. Okay, so they have a small hair-like structure which helps in trapping the pollen grains. These pollen grains, when they are land on the stigmatic surface, there are certain type of chemicals which help the pollen grain to germinate on the stigmatic surface, and germinated pollen tube now passes through the style and reaches the ovary, and inside the ovary there is a chamber. That chamber we call it as locule. Okay, so this is the ovary. Chamber inside the ovary is called locule. This long part we call it as style. Now question comes, why there is a need of having a style? Stigma, of course, it is a landing platform for the pollen grain, and it also helps in trapping the pollen grains, as well as chemical which is present in the stigmatic surface allow the pollen grains to germinate and to produce the pollen tube. Now style is just like a what we observe in human beings. We produce millions of sperms, and these sperm, they travel in the reproductive tract and then reaches the egg in the middle of the journey. That means isthmic junction. See here, style actually helps in finding out the best pollen grain, having ability to germinate fast and reaches the ovule and undertake fertilization. By this way, plants can select a best quality pollen grains which has a male gamete and helps in production of their offsprings having best genetic quality. Okay, so pollen tubes which are formed fast, they reach the ovule which is present in the chamber of the ovary, that is called locule, and fertilization takes place. Here we'll try to understand the parts, that is stigma, style and ovary. Along with that, I also explained their role in the fertilization. Now stigma serves as the landing platform. Of course we studied. The style is the elongated slender part beneath the stigma. The style length will vary from flower to flower. Okay, it is not same. In some flower style is very long, in some flower style is very short. So basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, that we call it as locule. See, this locule, sometimes it is 1, then we use the term unilocular condition. If it is 2, then we use the term bilocular condition, trilocular, pentalocular. So the number of locules will vary from flower to flower. The placentation is located inside the ovarian cavity. Here you can see, without placentation, these ovules cannot attach to the ovary. So there is one cushion-like structure inside the ovary which will help the ovules to attach, and that we call it as placenta. There is one definition for this. Arrangement of ovules inside the ovary is called placentation. Okay. Arising from the placenta are the megasporangia. Now this term is very important. Don't get confused. Megasporangium is different, megasporangia is different, megaspore is different. So likewise, you should be very careful while using these words. Okay, so megasporangia commonly called the ovules. Now megasporangia is nothing but what? Ovule. Why they have used the term megasporangia? Because it is plural. What is megasporangium? It is singular. Suppose ovary is containing only one ovule, then we can use the term megasporangium. Suppose ovary is having multiple ovules like this, then we use the term megasporangia. So which is representing what? Ovule. Now the number of ovules in an ovary may be 1. Okay. In which plants we observe this single ovule? The examples are wheat, paddy and mango. In exam, there is a high chance that suppose 3 or more than 3 examples are given, they take these examples and frame a type of questions like, which of the following example is the odd one? Suppose they have given 4 examples, wheat, paddy, mango and papaya. What it represents actually? One of the example is having multicarpellary condition and remaining 3 are having single ovule, single ovule is present, and here it is more than one ovule is present in the case of papaya. So that is how you can choose the odd one. Okay, so many ovules are present in papaya, watermelon and orchids. You know, in orchids they produce numerous ovules, thousands of ovules. Okay, hope this is clear. See, this is the picture. Just observe the first one. So this is the female reproductive structure of the hibiscus flower. Now if you observe here, this is stigma. How many are there actually? 1, 2, 3, 4 and 5. What they represent actually? There are 5 carpels. Now they are all fused here. Here they are all fused. Now tell me, whatever we are seeing here, is it a style? Actually, this style part, it is covered by a monoadelphous condition in real flower, but here they separated it. So this is the long style, and here comes the ovary. This is the thalamus on which ovary is located. Now here what type of terminology we can use? It is multicarpellary syncarpous condition. Okay. So this is the again one more example. Multicarpellary syncarpous pistil of papaver. In second year you study this example one more time. Papaver somniferum. Okay. So this is one plant. Here you can see the stigma, and it is a syncarpous ovary. Again, one more example for syncarpous. So in exam you should remember hibiscus is also an example for syncarpous ovary, and papaver is also an example for syncarpous ovary. Now if you observe this one, this is actually a ovary of Michaelia champaka flower. Here you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, like that, so many ovaries, and they are all not fused, they are all free. So which type of technical term we use here? It is a apocarpous condition, multicarpellary apocarpous condition. Now this is one ovule. This we call it as anatropous ovule. There are many types, orthotropous, campylotropous. So here one type is given, that is anatropous ovule. The naming is mainly based on position of micropyle and chalaza with respect to funicle. Okay, so here the ovule is completely bent like this, and micropylar region is near to the funicle. So we use the term anatropous ovule. So let us study this anatropous ovule in detail. Let me explain this particular anatropous ovule by taking this diagram in the next page. Megasporangium or ovule. Just observe this picture. Such a wonderful picture. You can see all the details. See this oval shaped body. This is the body of the ovule. This is attached to the placenta with the help of stalk, and that we call it as funicle. So stalk of the ovule is called funicle. Now funicle attaches to the body of the ovule with the help of hilum. Now this hilum is the junction between funicle and ovule. There is one strong supportive structure here. This is called raphe. Here not mentioned. Actually this strong supportive structure is raphe. And if you see the body of the ovule, here there are two layers. This is the integument. Okay, integument. This integument is interrupted in the two regions. One is at the extreme end, another here in the micropylar region it is interrupted. So this micropyle is the region where pollen tube enters the megasporangium and releases the male gametes. Opposite to this we call it as chalazal end. Very, very important. Chalazal end represents the base of the ovule. Okay. Now inside if you observe there are so many cells. What are those actually? Nucellus. This nucellar cells act as a nursing cells. Again they nourishes the embryo sac. Here there will be a sporogenous tissue again. They undergo mitotic division and form a megaspore mother cell. Now megaspore mother cell undergo meiosis and form a 4 cells. Out of the 4 cells, 3 will degenerate, and the remaining one, which is nearer to the micropylar region, will starts to grow and become the embryo sac in the later part of the development. So that we will study in detail in the next part. So here same thing is explained. Let us go with the one by one. The ovule is small structure attaches to the placenta by means of stalk called funicle. So very important part, because in competitive exam you may get many questions based on statement based. So thoroughly understand each and every line from this. The body of the ovule fuses with the funicle in the region called hilum. So body of the ovule fuses with the funicle. This part is nothing but the hilum. Okay. Now each ovule has one or two protective envelopes called integuments. Do you know what happens to this integuments in the after fertilization? They become seed coats. Okay. So integuments encircle the nucellus except at the tip where the small opening called micropylar region. So here if you observe, integument is present entirely, but it is interrupted in the micropylar region. Opposite to micropylar end is the chalaza, representing the basal part of the ovule. This is very important. What it represents, chalaza? Chalaza represents the basal part of the ovule. Enclosed within the integument is a mass of cell called the nucellus. Cells of the nucellus have abundant reserve food material. Why it has abundant reserve food material? Because it needs to nourish the developing embryo sac. Now located in the nucellus is the embryo sac or female gametophyte. So one more name for embryo sac is female gametophyte. An ovule generally has a single embryo sac formed from the megaspore. This is very important. This is called monosporic development. Now coming to the topic megasporogenesis. Genesis, something related to formation. Megasporogenesis means the formation of megaspore. It's a process. How megaspore is formed? See, previously I have shown you one picture, the structure of anatropous ovule. Inside I have shown you the region where nucellus is present. Now some of the nucellar cells, they enlarge and becomes the sporogenous tissue, and this sporogenous tissue undergo mitosis and produces the megaspore mother cell. Now megaspore mother cell undergo meiosis and produces the 4 haploid cells. In that 3 will degenerate and 1 which is nearer to the micropylar region develop and form a embryo sac. That is what explained in this. Okay. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. So one of the cell will retain and other 3 will be degenerated. It is a large cell containing dense cytoplasm and prominent nucleus. The megaspore mother cell undergo meiotic divisions. And what is the importance of megaspore mother cell to undergoing meiosis? Why it should undergo meiosis? In order to maintain the ploidy. Because when gametes are formed, they should be haploid. Only when 2 haploid gametes fuse, they form a diploid zygote. Otherwise what happens? You just imagine there are 2 gametes. They are not haploid, they are diploid. When they fuse, what happens? The offspring will be tetraploid, which we commonly not observe in the nature. So in order to maintain the ploidy, it has to undergo the meiosis, so the gametes will become haploid. Meiosis results in the production of 4 megaspores. Here also 4 megaspores are formed just like in microsporogenesis, but in microsporogenesis all the 4 microspores are retained, but here it is different. When the 4 megaspores are formed, 3 will degenerate and 1 will be retained. Now, in a majority of the flowering plants, one of the megaspore is functional while the other 3 will degenerate. So if you observe here, in a majority of the flowering plants they have used. That means not in all the plants, all the 4 will degenerate, but in most of the case, 3 will degenerate and 1 will become the functional megaspore. only the functional megaspore develops into a female gametophyte. Another name for that is embryo sac. So this method of embryo sac formation from the single megaspore term monosporic development, very important. So one of the characteristic feature of angiosperms, monosporic development. What will be the ploidy of the cells of the nucellus? Now tell me, what is the ploidy of the nucellus? Of course, it is a part of the plant body. It is diploid because it has not undergone any type of meiosis. Megaspore mother cell, it must be diploid because only when it is diploid, when it undergo meiosis, it forms the haploid cells. So the functional megaspore is, of course, it is haploid, and the female gametophyte is also haploid. Simply by giving this type of question, he is giving the hint that there is a high chance in the competitive exam they ask questions on the ploidy of the cells. Okay, hope this concept is clear for you. Now, formation of the embryo sac. Again, one of the most important topic. Let me take the picture for better understanding. From this, it is very easy for me to explain the megasporogenesis. Just observe the first picture. So this is the megaspore. This is one ovule. Here you can see the outer layer is nucellus. Inside there is one megaspore mother cell. Okay, this megaspore mother cell undergo meiosis. In the first meiotic division, it forms two cells, that is called megaspore dyad. Here you can see this is one cell and this is another cell. So this is the meiotic division first. In the meiosis second, it forms four cells. Now the cell which is nearer to the micropylar region will be retained and other three cells will undergo degeneration. Now this is the cell. Here we should start with the single cell. Okay, now this is the cell which is present nearer to the micropylar region. Now one nucleus which is present inside this embryo sac is actually a haploid nucleus. Now this haploid nucleus undergo first mitotic division. Remember, mitotic division, and form the two nucleus. Okay. Now again it undergo mitosis and produces four nucleus. And half of the nucleus they move towards one polar region and another half will move towards the another polar region. Now subsequent mitotic division forms four nucleus at one polar region, another four nucleus at the another polar region. So total, how many nucleus are present? 8 nucleus is present. How many mitosis occurred? 3 times mitosis occurred. Okay, so which type of mitosis? Endomitosis. Because mitosis, when it takes place, when one nucleus becomes two nucleus, immediately it is not followed by the cytokinesis. So we can say it is endomitosis. Now once the 8 nucleus is formed, the cell nucleus starts to form a cell wall around it. The cell which is nearer to the chalazal region will become antipodal cells. So they form 3 antipodal cells. This is nearer to chalazal region. Now the part which is nearer to the micropylar region will form 2 synergids and 1 egg cell. Okay, now these 2 synergids, they act as a cell which provide nourishment to growing egg. Okay, if you see here in the micropylar region, they just flipped it. Okay, here they have given micropylar region, now here they are mentioning micropylar region. Finger-like projections are present and they are called filiform apparatus. They play very important role. When the pollen tube reaches the micropylar region, male gametes, how many male gametes are present? 2 male gametes are present. They have to be guided properly towards the egg and the polar nuclei. That guiding is done by the filiform apparatus which is present in the micropylar region. Now male nucleus, male gametes, One will fuse with the egg, another will fuse with the polar nuclei. Now if you look at this complete embryo sac, what you can see, there are 3 cells in the chalazal end and they are called antipodal cells. Their functions are not clear. And here if you see a very large cell, here it has 2 nucleus. The name of the cell is called primary endosperm cell, or you can say polar nuclei. They are diploid. Here antipodal cells are haploid. All the 3 cells are haploid. Polar nuclei is diploid. Synergid is haploid. And what about the egg cell? It is also haploid. Now when you look at the complete embryo sac, how many cells are present? There are 7 cells. How many nucleus is present? 8 nucleus is present. There is one important aspect you need to remember here. Once the male nucleus fuses with the egg will be fertilized and develop into embryo. Now the ploidy of the embryo will be diploid. But there is one more male nucleus. When it fuses with the polar nuclei is already diploid. Diploid plus haploid, it will form a triploid structure that will become an endosperm. So whenever the question comes, what is the ploidy of the endosperm? It is triploid. What is the ploidy of the polar nuclei? It is diploid. So you can expect many questions which is related to ploidy of the cells from this concept. Okay. So same thing is explained here. One more thing I want to make clear. Sometimes they ask questions like, what are the egg apparatus? Egg apparatus means egg cell and 2 synergids. Together we consider it as a egg apparatus. Okay. So this is about the pistil, Megasporangium and embryo sac. Hope this concept is clear for you, because first concept, which is stamen, microsporangium and pollen grain, and the second concept, pistil, Megasporangium and the embryo sac, is the basic concept to study all the aspects related to this topic. Hope these 2 concepts are very clear for you. So in this process, in microsporogenesis and megasporogenesis, plant was able to produce functional male gamete and functional female gamete. Now the question comes, how these 2 gametes are fused? Because what we studied in lower classes is, in primitive plants, gametes are non-motile and sometimes they are motile. When they are released into the environment, like in water medium, with the help of current of water, these 2 gametes will fuse and fertilization will take place. Now this is the advanced plant. How these 2 gametes are come in contact and how they are undergo fertilization is the very interesting concept. Here pollination is the process through which male gamete can reach the female gamete. Now what is the definition of pollination? It's very simple. Transfer of pollen from anther to the stigma of the flower is called pollination. As simple as it is. Now how this pollination will be achieved? There are different types of plants, they adapted for different mode of pollination. It is not same in all the plants. If you observe in nature, certain plants they produce beautiful flowers. Certain plants they produce a very small flowers. Some plants they produce some smelly flowers. Some plants they produce fragrant flowers. Some they produce nectars. Why all these different adaptation is there? Because in the course of evolution, these plants got evolved for different types of pollination. Some plants depend on insects, some plants depend on wind, some plants depend on water, some plants depends on birds, some plants depends on animals. So in some plants, the pollination is done by human beings. You know, vanilla is the best example for that. So same thing we'll study in detail here. First we will study the types of pollination, and then we will study what and all are the biotic type of pollinations and abiotic medium where pollination takes place. Okay, so let us go with the kinds of pollination. Autogamy, geitonogamy, and xenogamy are the three types. Let us go in detail with the autogamy.